Question 2. Complete the table below to show the monomer, polymer, and type of linkage for each macromolecule. So this question worth 2 marks. So it means that for every correct answer in each row, the mark will be 1 mark. Okay. So for macromolecule carbohydrates, obviously the answer would be monosaccharide. Okay, monosaccharides, or maybe if you want to give the specific monosaccharides such as glucose, it can be accepted. And then for nucleic acid, the monomer is nucleotide as it says there. Okay, I'm just going to write the answer. Monosaccharide. So the polymer for carbohydrate is polysaccharides. So you can have several types of polysaccharides. You can have starch, which is the storage carbohydrate. For plant, you can have glycogen, which is the storage carbohydrate in animal, which is usually it is stored in the liver. And then you have the cellulose, which is the component of plant cell wall. And then you have the chitin, okay, which is the component of exoskeleton of insects or arthropods, right? And then for the nucleic acid, your answer can be polynucleotides. Okay, the monomer is nucleotide, so the polymer will be polynucleotides. Or you can give the specific polynucleotides such as DNA or RNA. And then for the type of linkage for carbohydrates, the linkage present is glycosidic bond. Okay. Glycosidic bond. And to know it, okay, so there are several types of glycosidic bond. You have to know which is one for glycosidic linkage. For example, the bond is present in the molecule of glucose, which attach together alpha glucose to alpha glucose. Okay, so the linkage between the alpha glucose in the structure of maltose is 1,4, or the monomers of glucose are attached together through the linkage of 1,4 in the structure of starch. Okay, in the structure of glycogen. And then the beta glucose are linked together through the linkage of beta 1,4 glycosidic linkage. Okay, in cellulose. Cellulose, the monomers is beta glucose. Okay, so the linkage between beta glucose and cellulose is beta 1,4 glycosidic linkage. And then as for the other one, which is 1,6. Okay, so 1,6 is the bond that links the branches, okay, that are present in the structure of glycogen, okay, and also in the structure of amylopectin. Okay, so the branches are held together by 1,6 glycosidic linkage. So please refer back to your notes to see exactly where does this different types of glycosidic linkage occurs. So you have 1,4, 1,6, and then the other one is 1,2. Okay, so 1,2 is the bond that is present in the structure of sucrose. Okay, sucrose, as you know, the monomers is alpha glucose that is linked together. That is linked to the structure of the next monomer, which is fructose. Okay, so glucose and fructose, the linkage is 1,2. Okay, in the structure of sucrose. Okay, so you have to know where does this linkages occurs. Alright, and then for the nucleic acid, there is only one type of bond that links together the nucleotides, which is phosphodiester bond. Phosphodiester bond. Okay, so next question, B. Figure 7 shows the tertiary structure of a protein. Okay, so as you know, protein has 4 levels of structure. The primary structure is where the amino acid are linked together, okay, through linkage. We call it as peptide bond. Okay, so linear sequence of amino acid that is linked together by peptide bond. And then you have this secondary structure where there is two types, which is alpha helical and also beta peptide sheets. Okay, so you have to take note that for the secondary structure, it still involves one polypeptide chain. Okay, so for beta glucose, it involves a parallel segment uh, of a polypeptide. Okay, parallel segment that are folded. Okay, so they are folded into beta peptide sheet. So this still involves one polypeptide chain. So the primary structure, obviously one chain, okay, one polypeptide chain. The secondary level also involves only one chain. So do not assume for beta peptide sheet it involves several different chains. Okay, so it still involves one polypeptide chain where segment of the polypeptide chain of the same polypeptide chain, they are parallel to one another and are held by hydrogen bond. Same goes to the helical structure. Okay, so the helical structure is held also by hydrogen bonds. Okay, and then for the helical and beta peptide sheets, okay, so you have to take note the hydrogen bonds occurs between the oxygen atom, okay, oxygen atom of carboxyl of one amino acid and the hydrogen atom of amino group of another amino acid. So as for the helical structure, it occurs, the hydrogen bond occurs every 4 amino acid, okay, along the chain. And then for the tertiary structure, okay, the tertiary structure still involve one chain, okay, one polypeptide chain. So, but for this time, for example here, okay, so you have one polypeptide chain that are folded into specific shape, okay, like this, for example. And then along the chain, you have, for example, the helical, okay, and then you have the ketone, and then you have the helical, and then you have the pleated, for example, okay, and then you have the helical along here, okay, and all the way through. Okay, so uh, as you can see in the structure of tertiary structure of protein, okay, so it involves the helical structure in the secondary level, okay, and then you also have the beta peptide sheet also occurs in the tertiary structure. Okay, so to hold this tertiary structure is due to the interaction of side chain. Okay, side chain. As you know, the amino acid will have side chain, which is R. So the side chain of amino acid, there are several types. Okay, you can have non-polar amino acid, where, for example, in the side chain you have the presence of methyl, okay, methyl groups, hydrocarbons, and then you have polar side chain. For example, in the polar side chain you have hydroxyl group, okay, present. Um, and then you can have also the presence of carbonyl group, okay. So the presence of this functional group in the side chain of the amino acid causes the amino acid to be polar. And then another type of amino acid, it can be acidic, okay, acidic amino acid, where in the side chain it has the functional group carboxylic acid, where if this amino acid is in aqueous solution, it will ionize into COO negative. Okay, and then another type of amino acid is basic. Okay? Basic amino acid, where in the side chain it has the amino group. So if in the aqueous solution or in the cell, so it will ionize to become NH 3 plus. Okay, so if for example along the chain here you have the amino acid that has the side chain CH3, and this one has CH3 also. So this creates, okay, hydrophobic interactions. Okay, as you can see, the amino acid is in the core of the protein. Okay, so it is isolated from the protein, and then so therefore the interaction present is hydrophobic interaction and also van der Waals interaction. Okay, and then the other type of interaction that can be present if the side chain here, for example, has the functional group hydroxyl, okay, and then this one has carbonyl, okay. So the hydrogen bond will form between the hydrogen atom of hydroxyl and the oxygen atom of carbonyl. So the bond will the bond form is hydrogen bond, okay. So the interaction between the side chain along the polypeptide chain, okay, along the one polypeptide chain will hold a specific shape in the tertiary structure of protein. Okay, so this can cause the protein to attain specific shape in order for the protein to carry out its function. Right. So if we look at in this diagram here, figure 7 shows the tertiary structure of a protein. So as you can see for A, so the side chain for this amino acid, let's say like this is amino acid 1, it has the hydroxy group, and then the other amino acid here, it has carbonyl group. Okay, so hydrogen bond will form between hydrogen atom and also oxygen atom, right? So therefore, identify bond A, it will be hydrogen bond, right? And then you can spell, so it is preferable to spell the word hydrogen, okay? Hydrogen bond, okay? Spell the hydrogen, please. And then for this, okay, for the next interaction, okay, as you can see, I'm just gonna explain to you, okay, the interaction. So the interaction form is between this amino acid that has hydrocarbons, okay, and then this amino acid also has hydrocarbons. So obviously the interaction uh, form is hydrophobic interaction. As you can see, this amino acid is of type non-polar. Okay, they are non-polar amino acid. Same goes to here, non-polar amino acid. So the interaction will be hydrophobic interaction. So as the amino acid is within the core of the protein, it is isolated from the aqueous solution, aqueous surrounding outside, outside of the molecule. Okay. And then, so you have two types, hydrophobic interaction and also van der Waals interaction. And for the next amino acid here, as you can see, the side chain has the S. S stands for sulfur. So initially, the side chain has sulfhydryl group. Okay. So in the formation of the bond between the sulfur, so the hydrogen will be removed. Okay. So you have the formation of a covalent bond. So between the sulfur and sulfur. So the covalent bond specifically is called as disulfide bridge. Okay. Disulfide bridge. Okay, so it is a type of covalent bond. And then for the next amino acid here, okay, so this is one amino acid where the side chain present is the functional group present is carboxylic acid. So it is ionized. It has a negative charge, and the other amino acid you have the amino group. So this is basic amino acid. So in the aqueous solution, it will be ionized to become NH3 plus. So the interaction form or the bond form will be ionic bond. Okay, so because the attraction between the negative charge and the positive charge, okay, will produce ionic bond. Okay, so those are the bonds that are involved in the tertiary structure. So you have hydrogen bond, and then you have disulfide bridge, you have hydrophobic interaction, and van der Waals interaction, and then the other one is ionic bonds. So those are the 5 bonds or interactions involved in the formation of tertiary structure of protein. And then the last level, okay, which is quaternary. Okay, so quaternary, for quaternary, the 4th level here, it involves several 2 or more polypeptide chains that are joined together to form a functional protein. So B, the answer will be ionic bond. Okay, ionic bond. You just write ionic bond. Okay, so next question, draw a dipeptide and label the peptide bond. Okay, so dipeptide, basically it involves 2 amino acids. Okay, 2 amino acids that are linked together with a bond, peptide bond. So in this case, okay, the question did not give a specific monomer for the amino acid, okay, monomer specific structure of amino acid. So you can draw any type any structure of amino acid, or you can draw the basic structure of an amino acid. So you have to know how to draw the structure of amino acid. You have to know. You have to memorize the structure, okay, of an amino acid. So my pen seems not working. Just a moment. Okay. So draw a dipeptide and label the peptide bond. Alright, okay, so you have to know the structure of an amino acid. So basically, an amino acid consists of uh, carbon, which is bonded to hydrogen atom, and then the carbon bonded to an amino group. So nitrogen, hydrogen, hydrogen, okay, and then the carbon here has the side chain. Okay, so you can just simply write R, okay, R1, for example, the first side chain of the first amino acid, and then the carbon is bonded to a carboxyl group. Okay, oh, alright. So initially, okay, so I'm just gonna draw the structure of the first amino acid. So because this is carboxyl, see, so COH, okay, and then another amino acid, which is hydrogen. You just repeat the drawing, okay, of the first amino acid. So C double bond O, OH. Okay, so the question just want you to draw the structure of a dipeptide. So in this case, you don't have to draw the individual amino acid. You just draw the product, which is the dipeptide. So I'm drawing these 2 amino acids to make it clear for you to understand. Okay, so in this case, to join together these 2 amino acids, so amino acid 1 and also amino acid 2, okay, so it involves the reaction dehydration or condensation, where in the process, okay, hydrogen will be removed. Sorry, oxygen here. Oxygen hydroxyl group will be removed. Okay, and also the hydrogen atom of the amino group will be removed, and a water molecule will be discarded from this molecule. So the product will be, you just join these 2 molecules together. So therefore the product is, okay, hydrogen, nitrogen, carbon. Okay, so this one you really have to take note. So carbon from the carboxyl group, so carbon is bonded to oxygen. Okay, so because you have removed the hydroxyl group, so the carbon will be bonded to the nitrogen of the next amino acid. So the nitrogen will be bonded to the hydrogen atom. Okay, carbon. You just copy back what the initial amino acid there. All right. So please draw your molecule clearly. Okay. So because I have not enough space here. Okay, OH. All right. So don't forget, okay, the side chain here. Okay. So the bond, and the second question is you have to label. Okay, you have to specifically point the where is the peptide bond. So it is between the carbon and the nitrogen here. Okay, so just label peptide bond. Okay, peptide bond. So this is the correct amino acid. You have to draw it properly to get the 2 marks. Okay, so 2 mark, 1 mark for the correct structure of the dipeptide, and then the second mark is the labeling of the peptide bond. You have to specifically point the location of the peptide bond. Okay, so if you were to, for example, to draw it wrongly, only one atom for example, one atom is missing. Okay, let's say for example you forget to draw a hydrogen atom, okay, that is bonded to the nitrogen there. So you draw it like this, one atom is missing, so you will lose 1 mark. Okay, you will lose 1 mark because your structure of the amino acid is wrong. Okay, so please check your amino acid structure properly after you have drawn the molecule. Okay, so next question C. Purines and pyrimidines are nitrogenous bases that make up 2 different kinds of nucleotide bases in DNA and also RNA. So you have purines and also pyrimidines. For purines, okay, examples of nitrogenous bases for purine is adenine and guanine. Okay, adenine and guanine. As for pyrimidine, the bases are cytosine, thymine, and uracil. Okay, so these are the 5 different nitrogenous base. Okay, that can be present in DNA and also RNA. So list 2 types of pyrimidine in RNA. So pyrimidines, okay, is obviously cytosine and uracil. If you were to answer uracil and thymine, so your answer will be incorrect because thymine is not present in RNA. It only present in DNA. Okay. So number 2, it says here if one strand of a DNA molecule has the base sequence 5 prime, okay, ATT, GCAT 3 prime. Okay, so write down the complementary sequence of the given strand. Okay, so as you know, DNA strand, it consists of double-stranded DNA molecule. Okay, so it is basically helical. Okay, helical like that. Okay, helical. So the 2 strands, okay, are held together by hydrogen bonds between the bases. Okay, between the bases. So the bases along the DNA forms hydrogen bond, and the base has to be the base that are present along the 2 DNA strand. They has to complement each other. Okay, they has to complement each other, or we call it as complementary base pairing. So if so for one mark you have to write down the complementary sequence. Okay, it use the word complementary because A will pair with T. Okay, G will pair with C, for example. 5 pairs with 3, 3 pairs with 5. Okay, so they complete each other, they complement each other. So in the second strand here, it will be 3 prime. Okay, A pairs with T, A, okay, and then C, G, T A 5 prime. Okay, so remember you have to write the directionality, okay, the number on each end of the strand also. If you were to forget, okay, or you did not write the number, okay, on each end of the strand, your answer you might lose 1 mark. Okay, so to be on the safe side, please, okay, write the numbers also. Okay, write the directionality, write the two ends of the DNA strand. Okay, so that one is worth 1 mark. Okay, the complete strand here, the complete sequence. Okay, and then the figure 8 shows a simplified diagram of a triglyceride. Okay, okay, so triglyceride is a type of fat. Okay, so you can have saturated fats or unsaturated fats. Okay, so for triglyceride, it has two components, which is glycerol and also 3 molecules of fatty acid. Okay, so X will be glycerol, obviously. Okay, glycerol. Okay, for 1 mark. Number 2, give the solubility of X before condensation to form a triglyceride. So condensation is the reaction that joins together the molecule of glycerol to the 3 molecule of fatty acid. So obviously the process of condensation in producing the triglyceride will remove 3 water molecules. Okay, so back to the question, give the solubility characteristics of X. X is, we know that it is glycerol. So glycerol, it is a type of alcohol. Okay, a type of alcohol where if you look at the structure of glycerol, okay, it has the presence of 3 hydroxyl group. Okay, so as you know, hydroxyl group, its property is polar. Okay, so polar, because hydroxyl group is polar, therefore any polar molecule, it can form or it can interact with water molecule. Polar molecule can form hydrogen bonds, okay, with water molecule. So therefore glycerol molecule will be soluble in water. Okay, so therefore the answer will be soluble in water. Okay, or you can also write it is insoluble in organic solvent. Okay, vice versa. Okay, so that is for 1 mark.